Hello. Please hit like button and subscribe my channel. Also press bell icon for future video notifications. Thanks.
Elisabeth Knust leads a team of researchers from the Max Planck Institute of Molecular Cell Biology and Genetics, MPI-CBG, in Dresden, Germany, who describe an essential role for one such metabolic pathway in maintaining retinal health under conditions of stress. They studied the classic Drosophila genes cinnabar, cardinal, white, and scarlet, originally characterized decades ago and named due to their role in eye color pigmentation, in particular the formation of the brown pigment of the fly eye. These genes encode components of the kynurenine pathway, whose activity converts the amino acid tryptophan by various steps into other products. In this study, the authors have highlighted the function of this metabolic pathway in retinal health, independent of its role in pigment formation. The kynurenine pathway is an evolutionary conserved metabolic pathway that regulates a variety of biological processes. Its disruption can result in the buildup of either toxic or protective biomolecules or metabolites, which can worsen or improve, respectively, the health of the brain, including the retina. Knowledge on this important metabolic pathway was recently extended by the research team, led by Elisabeth Knust, Director Emerita at the MPI-CBG, in their publication in the journal PLOS Genetics. Being aware of the remarkable conservation of this metabolic pathway and the genes that regulate it, they used flies as a model system to unravel the role of individual metabolites in retinal health. The researchers looked at 4 genes, cinnabar, cardinal, white, and scarlet, named after abnormal eye colors following their loss in flies. Since the kynurenine pathway is conserved from flies to humans, we asked whether these genes regulate retinal health independent of their role in pigment formation, says Sarita Heber, one of the lead authors of the study. To find this out, the scientists used a combination of genetics, dietary changes, and biochemical analysis of metabolites to study different mutations of the fruit fly, Drosophila melanogaster. Sophia Trakoff, a co-author, developed a method for the biochemical analysis of the metabolites of the kynurenine pathway. This allowed the researchers to link different metabolite levels to the health state of the retina. They found that one metabolite, 3-hydroxykynurenine, 3-OH-K, is damaging to the retina. More importantly, they could show that the degree of degeneration is influenced by the balance between toxic 3-OH-K and protective metabolites, such as kynurenic acid, kynurenine, and not just by their absolute amounts. Sarita continues, we also fed 2 of these metabolites to normal, non-mutant, flies and found that 3-OH-K enhanced stress-induced retinal damage, whereas kynurenine protected the retina from stress-related damage. This means that retinal health in certain conditions can be improved by altering the ratio. Furthermore, by targeting these 4 genes and therefore 4 distinct steps within the pathway, the researchers were able to demonstrate that not only the accumulation of 3-OH-K as such, but also its location in the cell and hence its availability in further reactions, is important for retinal health. This work shows that the kynurenine pathway is important not only in pigment formation but that the level of individual metabolites fulfills important roles in maintaining retinal health, says Elisabeth Knust, who supervised the study. She concludes, in the future, The ratio of the various metabolites and the specific sites of their accumulation and activity should be taken into account in therapeutic strategies for diseases with impaired kynurenine pathway function, observed in various neurodegenerative conditions. Thanks for watching. Please subscribe my channel for more videos.